Antimicrobial peptides, or AMPs, are a class of small peptides that play a crucial role in the innate immune systems of various organisms. They exhibit broad-spectrum antimicrobial activity against bacteria, fungi, parasites, and viruses. Of particular significance are marine AMPs, as marine organisms, such as fish, have a stronger innate immune system from being constantly exposed to a wide range of pathogens in the aquatic environment. Our study is focused on one such marine AMP, epinecidin-1 or EPI-1, isolated from the orange-spotted grouper fish. AMPs such as EPI-1 are cationic in nature, and the mechanisms of action involve selectively disrupting bacterial membranes, the ability to modulate inflammatory response, and promote tissue repair, making it a promising candidate for accelerating the wound healing process. This is an important cause as the management of chronic wounds, especially ones with underlying pathological infections, constitute a major therapeutic challenge as they prolong tissue injury, impair normal healing, and lead to the emergence of drug-resistant pathogens. For cases such as diabetes, HIV, and AIDS, this could have severe consequences, including fatality. Thus, identifying novel candidates such as AMPs like EPI-1 to heal wound injuries and infections is necessary to prevent the worsening of such chronic wounds. Our study hypothesized that EPI-1 will increase the proliferation and wound closure of human melanoma cells. To achieve this, the dose-dependent effects of EPI-1 on cell proliferation and its impact on wound healing behavior were investigated. Moving on to the methods, epinecidin-1 was prepared by removing TFA counter ions with the help of a displacement reaction involving HCl through lyophilization. The lyophilized product was then resuspended in PBS and used for all the cellular assays. Cells were cultured and treated with doses of EPI-1 ranging from 5 to 500 microgram per ml. An XDT assay was then performed to measure the rate of cell proliferation post 24 hours of treatment. Two doses, 50 and 150 microgram per ml, had significant increases in cell proliferation, and they were then chosen for the colony formation and wound healing assays. The former involved monitoring of colony formation in the EPI-1 treated cells for a span of 6 days, while the latter was performed by creating a wound in a confluent monolayer of cells and observing the wound closure over 24 hours. PBS was used as vehicle control and 10% DMSO as positive control for all the assays. The results from the study led to two important findings. Firstly, that EPI-1 treatment leads to increased cell proliferation. This result was obtained from both the XDT assay and the colony formation assay, as depicted in figures 1 and 2. Figure 1 shows that there was a significant increase in cell proliferation from doses 7 to 150 microgram per ml after treatment with EPI-1. And figure 2 shows that there was an increase in colony formation after treatment with 50 microgram per ml of EPI-1. However, the results from the colony formation assay was simply a trend and had no significant effects. In both cases, dose 50 microgram per ml showed the maximum effects. Secondly, the study led to another important finding, that EPI-1 treatment leads to increased wound healing. As seen in figure 3, EPI-1 had a significant increase in the migration of A375 cells after treatment with a dose of 50 microgram per ml for 24 hours. From the results obtained from the study, we can thereby conclude that EPI-1 dose-dependently promoted epithelial wound healing properties such as cell proliferation and wound closure under in vitro conditions, making it a promising candidate against chronic wounds. Moreover, to gain further insights into the antimicrobial properties of EPI-1, its effects can be examined against strains of bacteria, including drug-resistant bacteria such as MRSA. These effects can then be compared to synergistic effects of EPI-1 with antibiotics against these strains of bacteria to further support the idea that EPI-1 promotes wound healing in chronic wounds that are infected with drug-resistant bacteria. Further studies can also be performed to analyze the composition of EPI-1 after TFA removal. The effects of a known pure form of EPI-1 can be compared against the study to determine if TFA disrupts the effects of EPI-1. Finally, I'd also like to take this opportunity to sincerely thank Dr. Jessica Livingston, Dr. Michael Gibson, Crystal, and Marla for the immense guidance and support throughout the development and execution of this research project. Thank you.